Our last speaker, Marc Diamond, my colleague at UT Southwestern. Please.
I want to talk about our work on tau. We've heard a lot about Alzheimer's disease, and it is one of many different neurodegenerative diseases that are all linked to this one protein. I'd like to have my slides up too. I guess I have to advance them. There we go. I need to disclose two things. One is that I was involved with David Holtzman in developing a therapeutic antibody. I'm not talking about it. And also some of the technologies developed in our lab have been licensed to companies for use in drug development. I started off by saying tauopathies are diverse, and I want to just emphasize this point, and I'll come back to it at the very end. The bottom right, you can see a neuron that's filled with tau from an Alzheimer brain, but there are many different disorders where tau accumulates inside of nerve cells. And each of these disorders affects different brain regions. And we can think about, for example, in the case of Alzheimer's, pathology beginning in one discrete region, in the entorhinal cortex, for example. And then what's been known is that it follows a progressive pattern. And this is just a cartoon here, but I'm illustrating the idea that the diseases progress based on the way neurons are connected to one another. And this is turning out to be true for a lot of different neurodegenerative diseases. They seem to initiate in certain brain regions, and then based on the connectivity of the neurons, the diseases appear to progress along networks. And so if you imagine a bunch of different start sites, you can imagine how we can get to a lot of different syndromes, depending on where the pathology first begins. So what is this protein tau? It was isolated in the 70s at UCSF in the Kirshner lab because it's stuck to microtubules. And indeed, it will stabilize microtubules in vitro by binding to them. What was discovered subsequently is that there are multiple mutations in this protein, especially focused, but not exclusively, in this red domain that I call the repeat domain because it has 3 or 4 repeated segments. And those are dominantly inherited mutations. If you have those 100% of the time, you'll develop a neurodegenerative disease that involves the accumulation of tau. So it's clearly causal in neurodegenerative disease. There are additional amino acids. I just wanted to illustrate hypothetically with the green and blue dots, because I'm going to come back to this idea, that appear to play a critical role in allowing this protein to form ordered assemblies, or amyloid structures, that I'll highlight in cartoon form in a minute. But for most of the time in the literature, if you read about tau protein, you'll read that it's an unstructured protein. It doesn't have any definable structure. And what I want to emphasize in this talk is that is not true. And in fact, the protein probably has multiple distinct conformational ensembles, some of which can lead to disease and some of which do not. So this protein can easily be purified. It's thermostable if you make it in bacteria. You can purify it from a variety of different ways. We learned to do this in my lab when Bess Frost was there. Her name's at the bottom. And you can, if you, under the right conditions in a test tube, tau will self-assemble to form amyloid fibrils, the likes of which you can see either by electron microscopy on the left or atomic force microscopy on the right. And these have some similarity to the fibrils that are seen. Something bad is happening. Can you back me up? I'm not touching anything and everything's just, okay. I know, they look awesome, but they're advancing way too fast. Anyway, we began to wonder about amyloid assemblies and the diversity of those tauopathies. And one of the experiments that Bess did when she was in the lab was to form tau fibrils in vitro that had different conformations, and then allow those fibrils to interact with native protein. And what she saw was that depending on the conformation of the first form of tau, when that native protein interacted with it and added on to the chain, it would grow more of that same conformation. And depending on the initial conformation that the tau formed, and in this case I'm showing sort of a helical shape or a straight filament, you could amplify that structure based on its interaction with native protein. And we now know that this process happens not only in a test tube, but in cells, and indeed in the brains of mice and humans. That is to say, tau forms what we call strains, which are discrete amyloid structures that replicate faithfully in living systems, just like prions, and have unique biological effects depending on their conformation. And with 15 minutes to talk, I don't have a chance to do justice to a lot of the work that has gone into these ideas. But they're important things to remember. This one protein can assemble in different ways and give you different structures with different biological effects. So knowing this, we began really to wonder, could that progression of the diseases through brain networks actually involve mechanisms similar to that which had been defined in the last decades by Stan Prusner and others as they studied prions. And we began to wonder, and Beth was one of the first people I got to talk about this in my lab. I said, this is a crazy idea, but if it's true, it'll change a lot of things. And she was game, fortunately. But the idea was that in a cell in the brain, let's just imagine a single cell or a group of cells, if the tau converts to the abnormal form, and if by chance that tau could escape the first cell and gain access to a neighboring or connected cell, that process of in vitro amplification could happen potentially in a cell replicating particular structures. And this was an important new idea for us because it highlighted the importance not only of what's going on in the cell, but how an individual protein might exit that cell and move around. And that creates interesting opportunities for therapy, for example, vaccine therapies. So we've subsequently learned that depending on that conformation of tau that propagates from one cell to another through the brain, you will create entirely different neurodegenerative diseases. And we know this first from isolating distinct structures from the different human tauopathies, sort of illustrated in cartoon form here. But if we purify them to single conformers and inoculate mouse brain with, or not a living mouse, with these different conformers, we can create a myriad of different types of tauopathies that entirely is reminiscent of all the diversity seen in humans. So we believe that the diversity of all those different diseases that I alluded to in the beginning is actually traceable back to the that first seed, the structure of that first assembly. This basic observation that an aggregate can propagate pathology was first observed by Bess doing a really simple experiment. She had a cultured neuron, or neural cell, I should say, and full-length tau was expressed in that cell, fused to a fluorescent protein, and it would orient itself on microtubules. But if Bess added fibrils to the media bathing these cells, the protein on the inside of the cell would convert to a fibrillar form and accumulate in these inclusions. This was a very dramatic finding. It's easy to see this effect here, and it was very exciting to see it under a microscope. But it turns out that simple observation kicked off this whole idea, really, that tau could be propagating pathology. And we've turned a simple cell assay to very good use as a means to detect tiny amounts of pathological forms of tau. And we did this by taking the full-length protein and modifying it genetically, taking away the parts that weren't involved in forming fibrils, leaving just this core domain, the repeat domain, which we then fused to complementary fluorescent proteins, cyan and yellow fluorescent protein. And when these proteins come into very close proximity, they change their spectroscopic qualities, such that in a dish, if a cell is exposed to these seeds, they will come in and trigger a kind of catastrophic aggregation event in the cells, which we can then read out with a machine, either a flow cytometer or a specialized microscope. So we use these cells to monitor the presence of pathological forms of tau. And we were very interested in this question. What what makes a seed? What makes that first thing that can trigger the pathology? There are different models out there, and indeed, there are different ways that protein amyloids can form, most likely. But one prevailing model was that at the time was that tau would form kind of amorphous clusters, which would then, by virtue of their proximity, could somehow reorient themselves into an ordered structure, which could then act as a seed. So this implied that the seed was a multimer. But another possibility that we were interested in was, could a single protein actually change its shape and form the seed as a single unit, which would then self-assemble? And so we tested this by purifying tau from Alzheimer brain and from in vitro sources. And again, I'm summarizing a lot of work here. But basically, by immunopurifying the tau, we could then use, I need to go backward, let's see. We could use a size exclusion chromatograph, which is basically like a molecular sieve, which separates assemblies based on their size. And we were able to isolate fractions that contain single monomer, dimer, trimer, on up the size scale. And we then used that biosensor cell system that I mentioned to you to test these. And we made a remarkable observation, and that was that a single protein purified from an Alzheimer brain as a single unit had this seeding potential. It had this ability to form pathological structures. And that led us to ask in animals, when does this first appear? So we took a mouse that gets progressive tauopathy, and we killed these mice at 1, 2, 3, or 4 weeks and purified the tau from their brain, looking at different assembly sizes from 1 up to 10 or 20. We saw nothing in terms of seeding activity until 4 weeks of age, when all of a sudden the monomer fractions showed seeding activity, and that fell off as we purified fractions that would have had larger assemblies. In other words, the first thing we could see was the monomer in these mice. And this becomes more obvious if you just look at the monomer seeding in light gray versus the dark gray is the insoluble material, those fibrils that we're imaging and everything. Those occur late in life, at a year in the animals, but we could see the seeds as early as 4 weeks as a single protein. So what is the shape? What's the basis of this? Well, we used a method in collaboration with Łukasz Joachimiak, one of our faculty members, of cross-linking with mass spectrometry, where you can basically take a globular protein, and by adding cross-linkers, you can basically cross-link local structures or distant structures, such as green here. By chopping up the protein with a protease and then using a mass spectrometer to measure the masses of these cross-linked species, you can actually make inferences about the structure of the protein. And this data has led to models of how we get to 2 different structures of this protein that we thought was unstructured. In the one case, in the form of tau, which is not reactive and which we find in healthy controls, I want you to observe these green and blue spheres, which represent amyloid-forming amino acids. They're buried in hairpins in this protein. In the seed-competent form, however, the cross-linking data, when applied with molecular modeling, predicts that these hairpins are unfolded and these reactive amino acids are now basically solvent accessible. And this is just a model. It is not a crystallography structure. But I can tell you that there's a lot of biophysical data biophysical tests that we have done which entirely corroborate this model. I just don't have time to go through it today. So this is where we are now, to summarize. We think that these diseases are actually a result of a molecule, a protein, changing its shape from what we would call an inert form, where it's not reactive, to a seed-competent form. And what I haven't had time to tell you about is that within the seed-competent form, we think there are multiple substructures, each of which encodes the conformation of a larger assembly. The implications of this work are really profound for us, because it suggests that we should be able to, if we can detect the formation of these abnormal structures early, just from as we could see in the mice, when at 4 weeks of age we could see them, we should be able to detect incipient disease in people. And moreover, if we're clever enough to discriminate the distinct substructures that are present, we might even be able to say what type of tauopathy is brewing in an individual patient. So from a diagnostic standpoint, this is important. This also becomes very important thinking about therapies because we are now working on small molecules that stable the inert form, as well as immunotherapies that specifically target unique conformations that we think are propagating. So with that, I'd like to stop. I want to thank the many members of my lab who contributed to this project over the last 15 years and our funding sources, and I'm happy to take some questions. Thanks.
Thank you very much. We have time for questions. Yes, please.
Very nice. I'm just wondering if the transition from the normal to the seed competent form is being driven by any PTMs?
That's a great question. Do PTMs change the propensity? We think so. We think there are multiple ways the protein gets signaled to change its shape. One is phosphorylation. We've been able to do comparative mass spec of the monomer from different states, including in the mice over these 1-week periods, and we've actually identified residues that become phosphorylated when it has transitioned. We've then flipped that around and introduced those mutations, and it appears that they promote the conversion. We also think other ligands in the cell may promote that conversion as well.
And then, if I may, would those potentially be targets for prevention? Are the kinases responsible for those phosphorylations?
There's been considerable interest over the years in targeting kinases because it's well known that tau in its large fibrillar assemblies is so-called, they say, hyperphosphorylated, which just means there's a lot. And none of that has worked very well, bottom line. So I actually don't anticipate that that will be the best way forward. I think we can probably do better with some more specific ways to attack the conformations.
Okay, we're going to get a question here and then a question from over there.
Very good talk.
Thanks.
I have a question. Is there any linkage between events like traumatic brain injury, sudden mechanical jolt, concussion, that can lead to this tau protein accumulation, or even the amyloid accumulation, eventually can lead to AD?
Right. So the question is, if I can paraphrase, it's how could brain injury lead to this process starting? I can tell you we've collaborated with Anne McKee, getting brains from CTE. They clearly have what we would call tau prions that we can detect in these biosensor assays. The relationship of brain trauma and the cell stress involved with that and how that translates into a protein changing its shape, I don't know. We can make hypotheses that are probably testable about how that could work. But I do envision that there are many ways you can get to this shape change, this shape shift of tau. One could be some sort of cell injury. Another just could be time and sort of random conformational events, as well as other genetic factors. But yes, I do think that there's going to be a relationship of trauma to the protein changing its shape. We just don't have I can see the end product. I can't tell you the precise mechanism.
Okay, we'll take a question there, and then the last question will be from Lauren. But here, please.
Beautiful talk.
Thanks.
My question has to do with genetics. So there's going to be different alleles that are going to be present in different people, and those contain different amino acids that can give you different shapes of proteins. So, if you look at a really large data set, are there any shapes that you're looking at with the plaques that correlate to any kind of allelic variation?
Right, that's a great question, especially in light of what we just heard earlier from Chris Walsh, which I actually wondered about. Could somatic mutation lead to well, clearly we know they're in dominant cases, yes. Sporadic cases, I'm not aware, which doesn't mean it's not true, of people sequencing tau from sporadic AD just to determine, or is you asking a different question?
Alleles, variation in alleles from one person to another. I mean,
whether or not it's okay. So there are haplotypes of tau that are associated, but it's hard to relate them in the kind of way we'd like to in a biochemistry sense of a clear effect on shape change. Yeah.
Okay, last question from Lauren.
Lauren.
That really interesting stuff. It seems to me there's a potential contrast between what you're talking about in the first talk, where when we think about, for example, diagnosing Alzheimer's, it's still sort of fairly black and white, do you or do you not have the following things? And what you're suggesting is that there's profound diversity in the way that these molecular parts can potentially assemble and how they might spread and so on. So I'm just curious, in the long run, do you think it's going to be the case that diagnosis is going to depend on actually understanding the molecular structure of the amyloid, the tau, and everything else?
It's important to remember, so the question is, I've just talked about diversity of tauopathy, and yet we've been hearing specifics about Alzheimer's. Alzheimer's is by far the most common tauopathy. It's fairly uniform as far as we can tell in terms of the shapes of tau that cause it, as opposed to some of the other tauopathies where there is more diversity. In my fantasy world, we'll be able to take some spinal fluid from a healthy person and detect the tiniest amounts of these alternatively folded proteins, and by virtue of that, say you are on the road to Alzheimer's or you're on the road to progressive supranuclear palsy or whatever it is. I don't know if that's ultimately going to matter. If you have a great therapy, it may not matter that I can subtype diseases. However, I think the lessons from cancer biology tell us that that matters a great deal. And especially when you talk about these processes are evolutionary, basically different conformers could be selected for and against. We know that from the prion world. So I do think it will matter. I'm not sure how much it will matter, and that's just where we are now. Yeah.
Okay, thanks very much, Mark.